Hello everyone. Today we are going to start a new genital ulcer disease, Donovanosis. It will be covered in 3 parts. In this part, I am dealing with history, epidemiology, including agent and pathogenesis. So today is the first class for the topic Donovanosis. The bacteria Klebsiella granulomatosis, which was previously known as Calymatobacter granulomatosis, is the causative agent. It is a venereal disease, that is a sexually transmitted infection. The history of Donovanosis is very interesting. This is a monograph from WHO which was published in 1954 and it was written by Rajam and Rangia. It is also referred as granuloma inguinale or granuloma venereum. They have defined as Donovanosis is a chronic, slowly progressive, mildly contagious disease of venereal origin, and it is characterized by granulomatous ulceration of the genitalia and neighboring sites, with little or no tendency to spontaneous healing. The interesting part is it was first recognized in India by MacLeod in Madras Medical College. There are so many synonyms, but the most famous ones are granuloma inguinale, granuloma venereum. The others are like granuloma venereum genitoinguinale, granuloma genitoinguinale, infective granuloma, granuloma inguinale tropicum because it is found in tropical areas, chronic venereal source, ulcerating sclerosing granuloma, serpiginous ulceration of the groin, and ulcerating granuloma of the pudenda. If we go back in history, it was Charles Donovan who was a major. He first described the presence of certain intracellular bodies in the exudate of oral lesions. So this is interesting. An STI, but the causative agent first seen in the oral lesion may be due to the unnatural practices. Charles Donovan is the same person who discovered etiological agent of Kala Azar, that is Leishmania donovani, and these are the intracellular organisms which have safety pin appearance. We'll learn about in a few minutes. So there are other people like Pund and Greenblatt. They were the first people who gave the specific description of histology of granuloma inguinale, and they were the one who described these large macrophages which contained the bacteria in safety pin appearance. So they are also known as cells of Greenblatt or cells of Pund. Anderson was one who successfully cultivated Donovan bodies in the yolk sac of chick embryo. It was she who established the bacterial nature of the causative agent of those safety pin appearing things, which was confused, like misinterpreted by Charles Donovan as some parasite because he was from a parasitological background. He was a parasitologist. Later, Marmel and Santora coined the term Donovanosis. Now we'll move on to the epidemiology of the disease granuloma inguinale or Donovanosis. As already taught in the other 2 STI, gonorrhea and chancroid, that according to PSM or community medicine, there is agent, host and environmental factors for any disease. Host and environmental factors will remain same for all STI. So you may go back to chancroid or gonorrhea slides or class to go through it. I am not repeating it here. We will concentrate on the agent. The agent is Klebsiella granulomatosis, also known as Calymatobacter granulomatosis. So let me write it down. This is a gram-negative bacteria. Almost all STI, the etiological bacteria is gram-negative as far as I remember. This bacteria has two forms, the encapsulated form and the non-capsulated form. The encapsulated form is the mature form and the non-capsulated form is the immature form. So this, if we roughly draw, this is the bacterial body and there is a capsule of polysaccharide. This capsule is very important for the pathogenesis. So this capsule is very important. It helps the bacteria to evade the host immune system. Now there are also chromatin which can be at the center or at the periphery, or it can be distributed bipolarly, concentrated at the two ends of the bacilli. The non-capsulated form can be a coccus, it can be a diplococci, or it can be a bacilli. So in all, there are so many shapes of this bacteria, and this leads to the term pleomorphic. So if we see the chromatin, when it is stained, it gives a telephone handle appearance or a safety pin appearance, because the chromatin is concentrated at the ends, at the two poles of the bacteria. We just mark the structure, the polysaccharide capsule. This is the true capsule, and these are the pili or fimbria, which helps in the adhesion of the bacteria over the epithelial surface in pathogenesis. So now we'll write it gram negative. It is a non-motile and non-sporing bacteria, and it is a facultative aerobe. Strict human pathogen, and it exists intracellularly. That is why it cannot be grown on artificial media. They need yolk sac to be cultured, and the term pleomorphic is associated with this bacteria. So moving on to the pathogenesis, these bacteria first adhere via the pili over the epithelial surfaces and then slowly enter the mucosa or skin via the abrasions that occur in the intercourse. Now once these bacteria are inside the body, the neutrophils, which are the first defense cells, come to the site to fight them, and they are also seen as epidermal microabscesses. These neutrophils are seen in these microabscesses. Next, once the neutrophil is not able to contain the bacteria, the infection, macrophages come running. They engulf the bacteria, but since the bacteria has that capsule, they evade the phagolysosome fusion. So they cannot be fully destroyed. They cannot be destroyed. They multiply within the macrophage. They also resist oxidative killing by releasing antioxidants. Now these macrophages will release some pro-inflammatory cytokines, which are mostly these: interleukin 12, 6, 1 and tumor necrosis factor. These then in turn attract the CD4 cells. Basically the Th1 type differentiation is promoted. These CD4 cells will release their cytokines: interferon gamma, tumor necrosis factor, interleukin 1 and 2. Interleukin 1 and 2, it helps in T cell proliferation. So more of CD4 cells will be at the site, and interferon gamma attracts more macrophages. So I've simplified this diagram, but it is a vicious cycle and it is complex. So once these happen, all the cells aggregate. The macrophages might increase in size to contain the bacteria. They modify into epithelioid cells. So the epithelioid cells are there with the safety pin appearing bodies, which are actually the capsulated bacteria. Then these will be surrounded by lymphocytes and plasma cells. There can be there is also a lot of histiocytes. So there will be another fibroblasts also come into play, and so let me mark it first. These are the cells of green bladder pund, and the bacteria inside, which appear like telephone handle, are the Donovan bodies. Also let me draw some fibroblasts. These are the fibroblasts. So now, as you can see, it has formed a granuloma. These granuloma will form the subcutaneous papules, then nodules, and ultimately they will ulcerate and cause tissue destruction. So this was the pathogenesis, and I hope it is a bit clear for you. If you even write or draw these things a few times, you will be very confident on these. We'll meet in the next class with the clinical features of granuloma inguinale.